Dear viewers, greetings. In this video, we are going to see about nuclear magnetic resonance spectroscopy. In short form, it is called as NMR spectroscopy. This video is covering the following contents. First, an introduction about nuclear magnetic resonance spectroscopy, followed by principle of NMR spectroscopy, instrumentation of NMR spectroscopy, working procedure of NMR spectroscopy, steps in NMR spectroscopy, applications of NMR spectroscopy, advantages of NMR spectroscopy, and finally limitations of NMR spectroscopy. Nuclear magnetic resonance spectroscopy or NMR spectroscopy. Nuclear magnetic resonance spectroscopy is a technique used to study the magnetic properties of atomic nuclei. This NMR spectroscopy relies on the phenomenon of nuclear magnetic resonance, where certain atomic nuclei, when placed in a magnetic field, can absorb and emit electromagnetic radiations at specific frequencies. History of NMR spectroscopy. NMR spectroscopy was first discovered by Felix Bloch and Edward Mills Bussell independently in 1946. Both scientists are credited with the discovery of NMR as a technique to study nuclear properties using magnetic fields. In 1952, they both shared the Nobel Prize in Physics for their work on NMR. Principle of NMR spectroscopy. NMR spectroscopy is based on the interaction between atomic nuclei and an external magnetic field. The principle behind NMR is that many nuclei have spin and all nuclei are electrically charged. If an external magnetic field is applied, an energy transfer is possible between the base energy to a higher energy level, generally a single energy gap. The energy transfer takes place at a wavelength that corresponds to radio frequencies, and when the spin returns to its base level, energy is emitted at the same frequency. The signal that matches this transfer is measured in many ways and processed in order to yield an NMR spectrum for the nucleus concerned. Instrumentation of NMR spectroscopy. The instrumentation of NMR spectroscopy contains 7 parts. They are sample holder, permanent magnet, magnetic coils, sweep generator, radio frequency transmitter, radio frequency receiver, and readout systems. Sample holder. The sample holder is made up of glass tube with 8.5 cm long and 0.3 cm in diameter. Permanent magnet. The permanent magnet provides a homogeneous magnetic field, and magnetic coils or the coils induce a magnetic field when current flows through them. Sweep generator. The sweep generator produces an equal amount of magnetic field which pass through the sample. Radio frequency transmitter. Radio frequency transmitter is a radio transmitter coil transmitter that produces a short powerful pulse of radio waves. Radio frequency receiver. Radio frequency receiver is a radio receiver coil that detects radio frequencies emitted as nuclei relax to a lower energy level. Finally, readout system. The readout system is a computer that analyzes and records the data. Working mechanism of NMR spectroscopy. Sample tube. The sample, typically dissolved in a solvent, is placed in a tube and inserted into a strong magnetic field. Next, the magnet and magnetic field. A strong external magnetic field is applied to the sample, which causes the magnetic nuclei to align with or against the field. This creates energy difference between nuclear spin states, and which is essential for NMR. RF transmitter or radio frequency transmitter. The radio frequency transmitter generates radio frequency pulses which are absorbed by the nuclei in the sample. These pulses uses the nuclei away from their aligned position within the magnetic field. Absorption. When the nuclei returns to their original alignment, they emit energy. The absorption of radio frequency energy causes transition between the different nuclear spin states, and this process generates signals that contains information about the molecular environment of the nuclei. Detector. The emitted signals from the nuclei are detected with the help of detectors. The detector captures the NMR signal, which is called the free induction decay or FID. Printer. The data is processed usually via the Fourier transform to produce spectrum. The peaks corresponds to the different environments of the nuclei, providing information about the structure dynamics and composition of the sample. And here the magnetic controller. The magnetic controller adjusts and maintains the strength of the external magnetic field and ensuring the stability for accurate readings. Steps in NMR spectroscopy. There are 6 steps in NMR spectroscopy. The first step is sample preparation. The sample is usually dissolved in a diluted solvent to avoid interference from hydrogen atoms in the solvent. The sample is placed in a thin-walled NMR tube. The second step is sample placement in magnet. The NMR tube is inserted into the probe, which is located inside the magnetic field of the NMR instrument. The third step is RF pulse excitation. A short RF pulse or radio frequency pulse is applied to excite the nuclei in the sample. This pulse uses the nuclear spins, aligning them temporarily against the magnetic field. The fourth step is relaxation and signal detection. As the nuclei relax back to their equilibrium state, they emit radio frequency radiation. This emitted signal is detected by the receiver coil. The fifth step is Fourier transform. The detected signal is processed by a computer which uses a Fourier transform to convert the time domain signal into a frequency domain NMR spectrum. Finally, the step 6 is spectrum analysis. The resulting spectrum shows peaks which are corresponding to a different nuclear environment in the molecule, which can be analyzed to determine the molecular structure. Applications of NMR spectroscopy. In structural elucidation, NMR is extensively used in organic chemistry to determine the structure of organic compounds by identifying the number and type of atoms and their connectivity. In conformation and dynamics, NMR can provide information about molecular conformations and dynamic processes such as molecular folding or exchange phenomena. In pharmaceuticals, NMR is useful for quality control, drug design, and determining the interaction between drugs and biological targets. In metabolomics, NMR helps in identifying and quantifying metabolites in biological samples by providing insights into metabolic pathways and disease states. In polymers and material science, NMR can characterize polymers, determine monomer composition, and study the dynamics of polymer chains. In protein and nucleic acid structure, in biochemistry, NMR is used to determine the three-dimensional structure of proteins, DNA, and RNA in solution. Advantages of NMR spectroscopy. The first advantage is non-destructive. NMR spectroscopy is non-destructive, preserving the integrity of the sample for further analysis. The second advantage is detailed structure information. NMR spectroscopy provides detailed information about the structure, conformation, and environment of molecules, including stereochemistry and molecular dynamics. Quantitative analysis is the third advantage. NMR spectroscopy can be used to quantitatively analyze mixtures without extensive calibration. The fourth advantage is no need for crystals. Unlike X-ray crystallography, NMR does not require the sample to be crystallized, making it ideal for studying molecules in solution. The fifth and final advantage is versatility. NMR can study a wide range of samples, including small organic molecules, polymers, proteins, and nucleic acids. Limitations of NMR spectroscopy. The first limitation is sensitivity. NMR is less sensitive than other techniques like mass spectrometry or UV visible spectroscopy, particularly for nuclei like 13 carbon or 15 nitrogen, which have low natural abundance. The second limitation is sample size. NMR typically requires a large amount of sample, particularly in milligram quantities, compared to the techniques like mass spectroscopy, which can work with nanogram amounts. The third limitation is expensive instrumentation. High field NMR spectrometers are expensive and require cyanogens like liquid helium for the superconducting magnets. The fourth limitation is long acquisition time. For samples with low concentration or nuclei with low sensitivity, NMR experiments can take several hours or days to acquire sufficient data. The final and fifth limitation is limited to small to medium sized molecules. High resolution NMR is mainly used for small to medium sized molecules up to 30 kilodaltons. Larger molecules like proteins over 100 kDa are difficult to study with conventional NMR spectroscopy. Dear viewers, that's all about the nuclear magnetic resonance spectroscopy or NMR spectroscopy. Thank you for your support. Thank you.